have these opportunities to present our latest finding, the AI-driven target identification for ARS. Yes. Okay. I hope everyone can see my screen. Okay. If there's any problem, just feel free to let me know. Okay. Good. So I will start. Okay. So basically, I'm currently leading a team of application scientists working in Insilico medicines in Hong Kong. I'm the head of Hong Kong office. And my team is working on the AI-powered target discovery. So before that, I will quickly introduce my company a little bit. So basically, we are an AI drug discovery company and founded in 2014. Now we have over 200 employees over the world. We have offices over the world in US, Europe, Asia. So far, we already raised over $380 million since 2013. Okay, so our main technologies is from the GAN generative adversarial networks and deep reinforcement learning. Okay, so basically we are the first one to use the GANs for the to generate the novel molecules to tackle different targets. So basically our fully integrated AI platforms will have the Pandomics for target discovery and Chemistry 42 for novel target design and for novel small molecule design for different targets. And also we have the in-clinical to predict the clinical outcome. We are very well recognized in the industries. According to the MIT Technology Review, we are recognized as the one of with the fastest drug discovery platform. So, and then for our industries, basically, the drug discovery is a very it takes a very long time, over 10 years, and it costs more than $2 billion maybe. Yeah. So why? Because the successful rate is very low. Yes. And from the target discovery and then to the clinical phase, it takes a very, very long time and a very low successful rate. So that's the main problem here. And then in 2020, we try to think whether we can use AI to come up with some novel targets and novel molecules and for broad diseases with no cure and also associated with aging. So actually, ARS is one of our interests, yeah, because currently there is still no cure, right? So this is why we did a project on that before. So basically, it's to improve the efficiency of the drug discovery. We developed a fully integrated end-to-end drug discovery platform known as the Pharma AI. So there are three parts: pan-omics, this is used for the target discovery and omics data analysis. Chemistry 42, once we identify the targets, and then we can use Chemistry 42 to do some de novo small molecule design to tackle the targets of interest. And finally, we have the in-clinical program to help us to further to predict the clinical trial outcomes based on our target choice and on our small molecules as well as the clinical trial design. So we have already have a very successful program, the program 1 for IPF. We identify antifibrotic small molecules to tackle IPF. Okay, so basically from the target discovery to reach the IND enabling, it just took us 18 months. So it is definitely industry record. And then we already reached the phase 1 in less than 30 months. Yes, so it is very quick. Comparing to the traditional ways, it will take more than 4.5 years. So at least, and it will cost a lot of money. Yes, but we just did it very quickly with our Pharma AI platform. So it is not only the example. So in the last 1.5 years, we already announced the 8 preclinical candidates using our Pharma AI platform. And the latest one is the BCC we announced in May to tackle the COVID-19. So now I will come back to our platform. So basically, why we can do that? Yes, because we have a very comprehensive database. Because we have the, for example, the omics data. We have over 10 million samples, and they are come from the microarray, RNA-seq, methylomics, and proteomics data. And in terms of the chemistry, we have over 1 billion structures, which is predicted with the experimentally measured properties. And then in terms of the text database, we not only can include the publications, but also the grants, patents, clinical trial informations, as well as the financial reports as well. So here is the overview of the panoramics. As you can see here, there in panoramics, we have over 20 AI and bioinformatics model here to help us to rank the target and disease association. The most important part is the omics AI scores. As you can see here, we have the 13 AI models, the network neighborhood, mutated submodules, causal inference model, overexpression and knockout model. We use the length database as a reference. Mutations, we use the GWAS and TWAS data. Pathway model, we use our proprietary iPanda pathway algorithms, and then the reference, expressions, and matrix fertilization, and the heterogeneous graph walk. The last two model has the strongest AI components. Yes, we just launched it last year. Okay, and then we still have some traditional models, for example, the text-based AI scores and KOL score, and as well as the financial scores as well. So overall, our platform provides a very comprehensive analysis of the target. So we can find out the best genes that is highly associated with the particular indication that you are interested. And then we can go through a series of the druggable filters, druggability filters. For example, let's say if you are interested on a small molecule, you can turn on the small molecule filters to find out those small molecule targets. Or let's say you want to find out the antibodies targets, yes, sure, you can do that. And also you can filter them by different developmental level and tissue specificity as well. So I guess everyone may ask, so where is our AI component here? So what exactly? So basically, AI is used to identify the genes and diseases compound between the biological process and the association between them in the text. And recently, just this week, yeah, should be Monday, we launched the Pharma AI as well as the Pandemic 3.0 version. Okay, so we introduced a transformer-based knowledge graph that you can see that how we use it to help us to identify the genes, disease, and drugs connections. And also, very important, AI-driven graph-based model are used for ranking the target and genes associations. And last but not least, AI is also used to analyze the potential for the attention of the initiations of the phase 1 trial. So as there we have over 20 models now, I just had one. For example, here we use the interactive community as an example here. We try to collect the information of a particular genes or protein, and then put that together with all the information of a particular disease on this single bipartite graph. And then we try to see how well the genes and disease are being connected to each other. And then we do the pair embedding. We use the multi-layer perception MLP as the decoder to decode this pairing. So each of the genes has our scores, so that we can, based on that scores, to rank the target genes association. So that is one of the model. And then we need to test whether that model can perform very well or not. So we have to make sure it can really do very well with our backtest. So in that case, let's say, for example, we choose 2010 as an example. We try to collect the data before 2010 and then put that into the DAT model to predict the targets coming out into the market in the next several years. For example, 2012 and 2015. So after that, if the model, let's say we predict 20 targets, if 18 of them coming out into the market in the next several years or entered in the clinical trial phase 1 or passed to phase 3 or phase 2, and then it means that our model performed very well. Otherwise, we will further to improve our algorithms. So this is the basic idea. So each model will have its own validation matrix. And then x-axis show the target prediction enrichment, y-axis shows the statistical significance level. And then so score 2 perform much better than score 1. So this is why we choose score 2, the model, to help us. So what is the uniqueness of our approach? So basically, as you can see, we have a very, very comprehensive database that can really help us to find out the targets. It is very well curated for target discovery. And then we have the unique and comprehensive validations with the time machine model to prove that our algorithms can really help us to generate some truly novel hypotheses. This okay. And lastly, most of these AI scores are calculated in real time. So it is not just a database showing you, okay, A is connected to B. No, this is not just a database. It really depends on the data set you select and input in order to the system to do the analysis. Okay, so over the years, we published many papers, yes, because today is academic sharing. So back to 2016, we published the first paper on Nature Communications on our iPanda pathway analysis tools. It proved that our pathway analysis tool can really outperform the other pathway analysis tools. And then over the years, we developed it from these iPanda algorithms to an industrial scale target discovery platform, Pandomics. We try to, over the years, we published it over 18 papers, and after the product was launched, we used it. We published the paper aging at ARS, and ARS is the my focus today. So basically, ARS, the beauty of this paper is that we not only use our platform to propose some target, but at the same time, we collaborate with the biggest player in this field to validate them. So ALS currently is still a no-cure disease, and the mortality is very high, over 80% within the 5 years and 92% within the 10 years. Okay, the mean survival time is only 35 years. Most of them are classified as the sporadic ALS, and the familiar ALS is only less than 10% of the whole ALS populations. So Neuroscience is currently the approved drugs, but it only prolongs the patient's survival. It cannot cure the disease. So far, there's no cure. So this is why we try to think whether we can use AI to find out some new targets to tackle this disease. Yeah. So basically, we collaborate with the biggest player in this field. We got the iPSC data from ALS, and then we work with the professor from Hopkins, Harvard, Tsinghua, and Mayo Clinics help us to validate them, validate our targets with their Fry model. So here is the a detailed experiment of the study design. So basically, you use the publicly available data set that is available on Pandomics. Most of them are the postmodern CNS tissues, and then we upload the proprietary data from the ALS, the iPSC derived neural data, and then derive them into the familiar and sporadic ALS and put on the platform Pandomics to do the DEG analysis, pathway analysis, and target ID. And then after that, we identify some novel targets that we can use for novel drug discovery. And at the same time, we also find some targets that is already have some drug available. We can do drug repurposing. Yeah. And out of this, we can identify, we can further validate our targets with the Fry model. So this is the overall idea. So as you can see here, we use the Pandomics over 33 models to help us to identify the targets for different subgroup of the ALS. So here are the detailed informations. As mentioned before, let's say my group focus on the small molecule targets. So first of all, for the high confidence targets, I have to make sure it is accessible by the small molecules, relatively safe to use, and the most important, I have to make sure it is druggable. So I turn on the druggability filters because I'm not going to find some targets is only associated with the disease, but we really want to find some actionable targets. Okay, so I have to make sure it is druggable, and at the same time, I will, for the high confidence targets, we'll turn on all the omics model, text-based, because I just want to find something good for them. So here is the heat map. As you can see, these targets will combine all the omics scores and the text-based and financial scores, as well as the heat map will show you its druggability. For example, whether this target is accessible by the small molecules, antibody, its safety, whether it is novel, whether it already have the structure, the developmental level is what, and what is the target family, something like that. So basically, the system will add all these targets, but these subscores together to give you an overall ranking. And then for the novel target identifications, now we will relax the small molecule requirements. Again, make sure it is safe to use, relatively novel, must be druggable, because we are going to find something new. So in that case, we do not need the literature-based scores. So I will skip all these scores. Okay, we try to find out the new targets simply based on the omics scores. Okay, this is what the result and our philosophy. As you can see now, you have a list of completely new targets, and they have never been reported from the ARS before. So at the same time, they have a relatively good performance in terms of each of the omics model. And then they also fulfill the druggability requirement, for example, safe to use, with some of them with the structures acceptable by the small molecules. So that's the way how we find out the targets. And the next, based on that part, we proposed the 28 target for LS. This includes from different subgroups, for example, the high confidence target, novel targets, from the familial ARS subgroup, sporadic ARS subgroup with their protein family enrichments, something like that. Yeah. And then the next step is we try to validate them with the Fry model. So we use the C9 Fry models. For the Fry, they carry this G4C2 repeats. They will have a Fry ID generation, something like this. So this is the white top. Okay, this is the with the mutants. Okay, so this is model is pretty well accepted in the field. So actually, Professor Zhang, one of our collaborators, already published a paper in Nature 2015. He identified some targets that can rescue the fry eye. Well, one of the famous targets for ALS is the VCP. And then we use this model and also work with the same person, Professor Zhang, to further validate our targets. And then here is the very impressive results. Okay, so the fry carry the mutants will have a fry eye degeneration like this one. And then if there's then no mutants or there's no degeneration, this is the wild type. We can take it as this one. As you can see, out of the 26 homologs of the fry eye, we use the RNAi to inhibit our targets. As you can see here, the results are very impressive. Once we inhibit the expression of our targets, and then it can really help to rescue the neurodegenerations in the fry eye. So the results are very impressive. And most important is that 8 of these genes are unreported. Okay, why we say 9 here? Because okay, I can show you. One of them is the VCP, which is already reported by Professor Zhang in 2015 in his Nature paper. So the other 8 are completely new. So the beauty of this research is that it highlights we can really use our AI target discovery platform to identify some truly novel targets, at the same time can validate it. So it is very impressive. And then besides the targets, we also, because as I mentioned, our pathway analysis is very good. And we also try to compare the dysregulated pathway in the CNS samples. As you can see that most of the especially the familial ALS patients, they have a dysregulated pathways in the immune system, cell cycle, programmed cell death, yes, and unfolded protein, something like that. And then, but in contrast, as you can see, for the sporadic ALS patients, they have some dysregulated pathway in other cellular process, for example, the RNA polymerase 3 transcriptomase, or even the signaling by FGFR. Okay, so overall, we can see that only few dysregulated pathways are overlapped between the familial ALS and the sporadic ALS. So it is quite interesting. Maybe it is that's why actually they are very different actually. Yeah. Okay. So and finally, we summarize all our targets in this diagram, and hopefully it can help to further to advance the ALS study. So we also provided the full scheme and our rationale, our target discovery approach on this ALS.ai. So feel free to visit it. And last but not least, today I introduced how we can use pandomics to find out some good targets for ALS and further validate it. After that, we can further use our Chemistry 42 program as well as the in-clinical program to further speed up the drug discovery for ALS. So this is what I call it a fully integrated AI platform for drug discovery. Okay, so because of the time is very tight, so that's all for me today. So if you have any questions or you have any interest on our technologies or our studies, just feel free to contact me. Yeah, thank you very much. Yeah, thank you.